Continue that. Is my screen visible?
Yes, sir.
So did you follow up the session from the YouTube channel? So because this glycolysis that I've been teaching it, it's for it's in depth. Like it's not just the normal process. It's at the cellular level. So did you check up on that, or should I just?
Sir, actually, I just went through it pretty quickly, so I was not able to go through the details of it in your lecture recording.
So I'll give you a brief introduction, whatever I have taught in last two classes, and then we'll proceed with that, from that. Okay.
Okay, sir.
So first thing, we already know that glycolysis is basically breakdown of glucose to pyruvate thing, and then after that etc continues, and then we get a lot of energy from that. So okay. Okay. So have you heard about blood transporters, like glucose transporters?
Yes, sir. They are in intestine and also in liver.
So there are how many types of glutes?
So I'm aware of GLUT2 and GLUT3.
You are aware of GLUT 1 and GLUT 3. So you are aware of. So first thing is there are around 14 types of glutes, out of which GLUT 1, 2, 3, 4, 5, and 7. They are the most important one. Like the rest of them is not that thing. So you know about GLUT 2 and 3. So where do we find GLUT 2?
So is it in intestine?
Okay.
After the breakdown of the food, eating the brush border cells are the terminal uh, point from where the carbohydrates are absorbed.
Okay, so okay, first answer is what is the specialty of GLUT 2?
Sir, actually, I prepared for oxidative phosphorylation, but I will just tell the very general thing that I know. Glue transporters are secondary active transporters, secondary active pumps where we use sodium along with glucose to take the glucose inside.
Okay, you mentioned secondary active transport. Active transport, okay. So you mentioned about sodium, glucose, co-transport. Okay. So first thing that about GLUT2 is it's a two-way transporter. Except this GLUT2, none of the glutes out of 14, none of them are two-way transporters. For example, okay, first let's start with this. I'll give you a general thing. For example, GLUT1, it is present all over the body. It's ubiquitous. Every other cell in our body will have this GLUT1, because mainly RBCs and brain cells. GLUT2, it is one of the specialties that it's a two-way transporter. Like every other glutes, 1, 3, 4, 5, 7, everything, they are only the single way. They can only either let in the glucose or let out the glucose. So, but this one has a specialty. It can take in as well as take out. So basically, GLUT2 is like this. It can take out as well as take in. So this GLUT2 is special, and it is present in liver and pancreas. And GLUT3 again is ubiquitous. GLUT4, this is one more special GLUT. It is mediated by insulin. So every other cell, that is where glucose is stored. What are the places where glucose is stored?
So in form of glycogen, it is stored in muscles as well as liver.
liver,
hepatocytes.
then muscles, and like they're basically SKMs. So this is liver, skeletal muscles, and adipocytes. And glucose 5, glut 5 is basically present on our what? It's on the sperm. And the specialty of glucose, glut 5 is basically, and testis as well. Is that it transports not the glucose, it transports the fructose. And then glut 7 is present on endoplasmic reticulum. This is involved in gluconeogenesis. Okay, like I'll give a general thing here. Okay, so let's start with here. Actually, like I had to finish this glycolysis first, and then the plan was to go to the oxidative phosphorylation later. So first I'll complete that. So just because you don't have the idea that we in the last class that I taught, I'll give you a brief introduction. For example, okay, let's start with when you eat your food, so it basically goes from this is a GIT tract. Do you know what is GIT?
Yes, the gastrointestinal tract starting right from the mouth, buccal cavity to anus.
Mm. So this is our that like we intake our food in complex carbs, and here the food gets broken down. So what are the what are the types of monosaccharide that we get it? Like we get from the breaking down of the complex carbohydrates. One is glucose for sure. Then
So fructose, mannose.
the monosaccharide mostly like glucose, fructose, and galactose is the major three monosaccharide that has been like is utilized, and then this from here it passes out and then it goes to liver. Here via the portal vein. Here via the portal vein, this all the monosaccharides are shifted from this GIT tract to the liver, and here liver. Uh, what are the liver cells called?
Hepatocytes.
So hepatocytes. So in liver, as I mentioned there, like GLUT2. So here, like when we eat, when we are on a heavy diet, like when we just had lunch or something, there is a like higher, like larger influx of carbohydrate in our body, and then this carbohydrates are broken down into glucose, fructose, and galactose, and from that, from this GIT tract, via the portal vein, it goes to the liver. Now, it can't. This liver is the regulator here, like master regulator here. It regulates the intake and outtake, like how much it should store and how much it should release in the blood. So this glucose, when the glucose comes in here, like all the three forms, glucose, fructose, and galactose, all these three forms, they either gets like they get stored in glycogen form or are like released in the blood, like blood vessels, like in the vein. This is our vein, blood vessels, blood vessels, released in the blood. So here the what I said. So here, when the this monosaccharide reach the liver, they are either are like stored in the form of glycogen, and after that, it's small amount, like some amount of glucose based on the requirement is released into the bloodstream, and then from this bloodstream it goes into the cell. Now, like from the bloodstream, there is this interstitial fluid, like interstitial space here. It goes into that, and then from there it goes into the cell. Now this glutes, that is a glucose transporters, that's why like they play a role here, like how this glucose will go into this cell. So they are going with the help of this glucose, so gluts. And when for example, there are different cell types. When that's liver, as I told you, this glut 1 and 3 are ubiquitous. Every other cell in our body will have glut 1 and 3, but this special cells, that is SKM, skeletal muscle cells, liver, and adipose tissue, they will have a um, special glut, that is glut 4. It is because it is activated by insulin. As you all know, that when we intake lot of carbohydrates, our insulin goes up, and then this insulin directs us to take the glucose, since there is a like lot of glucose in our bloodstream. In order to maintain this sugar level in the glucose, sorry, sugar level in the blood, this insulin is released, and this insulin goes and binds to the receptors on the cell, and it tells the cells that there is like lot of glucose in the blood, please take it in. So basically here, like this is the mechanism. If this is a cell, so as I told you, GLUT1 and 3 are commonly present. So every other cell will have this thing. So this is GLUT1 and GLUT1 and 3. Now, um, and normally in your blood, in a cell, these glutes will be inside the like GLUT4. They are like very special type of glucose transporters. They will be inside a membrane wall. Like they are inside the bound in the vesicles, and they will be inside the cytoplasm. So these are the GLUT4. This is GLUT1 and 3, and these are GLUT4. So do you know what type of receptor is insulin receptor? So it's a receptor tyrosine kinase. Like it's a special type of receptor family, membrane bound receptor family. So when there is a like lot of glucose in your body, insulin is released by the pancreas, and this insulin will go and bind to the certain cells, mostly this SKM, liver and adipocytes. And when this insulin binds here, it will give a signal here, and due to these signals, this vesicle containing GLUT4, it will go and fuse with the membrane, and this membrane then will have the this membrane will now have the GLUT4, and then abundance of glucose will be taken inside this. The specialty here of glucose 4 GLUT4 is because it can take a higher, like a larger abundance of glucose inside in one go, while this GLUT1 and 3, they just have work on the basal level. The affinity is like quite high. Okay, so here's a, like one thing. As I told you, GLUT1 and 3 are present on the like they are ubiquitous, like they are present in every other cell. But GLUT4, they can take like larger amount of glucose inside, but they are not present on this cells like brain and RBCs. Like why do you think like it is a like it? Why do you think it does not have the GLUT4?
So maybe because the excess of glucose can be converted to the storage form in the cells that you have mentioned.
But they don't have the enzymes, right? Like glucose, like RBCs and brain doesn't have the it. It is only stored in SKM and liver, and they have the enzymes to store that. So first thing is when in your body the glucose amount is quite low, like I mean like very less, and like for example you're in fasting state, like you haven't had your food like in a like I mean a day or two. So even when your body doesn't have any glucose, doesn't have any energy, it's still like your blood's like RBCs and brain needs to function. That's why this GLUT1 and 3, since the glucose is low, there will be no insulin. So when then there is no insulin, GLUT4 won't be getting activated, and therefore, in order to take that minimal amount of glucose that is in your body to keep you alive, like I mean to keep you in an active state, this 1 and 3 are present. Uh, this 1 and 3 are present on the like very important cell types of our body. The that is our brain and RBCs. So even when your body is in fasting state, even when you don't have this glucose in your body, it still has to take up the glucose in order to provide you the energy and being to keep you like active. and therefore, like as I told you, that even when the glucose is very low, it still has the ability to take this glucose inside. So that means its affinity is high or low.
Hi, sir.
Yeah. So the affinity of this glucose compared to glut 1 and 3 compared to any other glucose is very, very high. So even when the glucose is at like really limited amount, it still can take it in and keep it inside. While glut 4 won't get activated. It needs abundance of glucose, and it is only activated because of the insulin. So insulin is activating the glut4. So it is only present on skeletal muscle cells, liver, and adipocytes. Now, this is 1, 3, and 4. Now we were talking about glut2. So first tell me, why do you think, like I have told you one thing, that glut2 acts both ways. Like it can take in the glucose and it can take out glucose. So where do you think it will be present?
So maybe on the cells which are able to store glucose, and then when needed, the glucose glycogen can be broken down to release
Hmm.
glucose out.
So it is present on liver. It can like the hepatocytes here. They have this glut2 specifically. So therein they can, like once the glucose is very high amount, they can just take it in, store it in glycogen. And if the body needs this glucose, it can just take it out and then give the body the glucose. So this here, and any other cell type? So what is the cell type that produces insulin?
Pancreas, sir.
So this pancreatic cells, they have this glut2 because they need to be always be like for example, suppose this is a cell. So and this is the interstitial space between the blood vessels and the cell. Here is the blood vessels. So now when we receive glucose from the liver to this blood vessels and then to this interstitial space, it's just a normal diffusion. The cells basically move from higher concentration, like higher glucose concentration, to lower glucose concentration. So it's a basic diffusion here. There is no channels here. It's just a diffusion. And from this interstitial space to the cells, there is requirement of this gluts. So basically, it's a facilitated diffusion. So one thing is that glucose moves from higher concentration to lower concentration always. But there is this one special cell type, that is this pancreas. It does not, like it has to be in optimum level with that of the environment. So this, for example, this is the pancreatic cell. Pancreatic cell. So and this is the interstitial fluid. It has to be always in touch with, like if the glucose is high here, it has to be high here. If it is low, if it is then it should be low here. So in order to keep this homeostasis between this interstitial space and this pancreatic cell, the glut2 is present. There is no movement of glucose, it's just for the sensing. So glut2 in the pancreas is for sensing the glucose outside, in the outside environment. So when it senses whether if the glucose is very high, this will release insulin, and if it's low, it will release the glucagon thing. So pancreatic cells, so that's why this glut2 is present there. Now we have covered glut 1, 2, 3, and 4. Now glut 5. As I told you, this is special because it does not transfer glucose, it transfers the fructose. Um, so it is basically present in our testes and sperms. And glut 7 is present on um, endoplasmic reticulum, where gluconeogenesis, like if in a cell gluconeogenesis is occurring, it is occurring at the site of endoplasmic reticulum. That is SCR. Now this was the glut thing. Okay, now let's go with the glycolysis part. Now, for example, this is a glucose, and this is a cell. Normal cell. Like for example, let's think this as a SKM. So which kind of glute will be present here?
So 1, 2, and 4.
Yeah, 1, 2, and 4. So like here mostly like let's talk about 4. So now since the concentration of glucose is outside is very high and in the cell it's really low, based on the gradient it will move in. So glucose will move in. Now after a period of time, the glucose concentration in the cells will be very high. So like do you think this glucose will move back to the interstitial space? Now after that, like for example you just had your meal,
Yes.
the concentration of glucose is quite high in your blood and it will like based on the concentration gradient it's moving from the interstitial space to the cell. Now after a time like when a lot of glucose comes in, eventually like the concentration of glucose will be very high here and will be getting lesser here. So do you think this glucose will go out?
So how can it go out via 4, 1, or 3? Because they are not two-way transporter.
Mhm. So like if there is any other membrane, like if what do you say, if there is a possibility of going like if this glucose goes out, like for example if there is a liver cells glut2, so if they are not even storing it, if there is a concentration gets like quite high, so how do you think the glycolysis will proceed further? So why do you think like how to stop this glucose inside? That's my question.
So we change its entire conformation by attaching a phosphate so that it no longer can pass through the loop 2.
Yeah, so it will come and get phosphorylated, and this phosphorus, when the phosphorus gets bind, like why do you think it won't pass? If the like if the glucose gets phosphorylated.
So is it because change in the conformation and also phosphate is charged?
Yeah, like it is because this phosphate makes it this glucose very charged molecule. It becomes polar and it can't easily like pass out the cell. Okay, now we have heard about glucose, and this is glucose 6 phosphate. So what is the enzyme involved here?
Hexokinase?
Hexokinase. Any other enzyme?
Glucokinase?
And glucokinase.
Glucokinase.
So what is the difference between this hexokinase and glucokinase? I
So
guess.
I guess hexokinase is present in all the cells and glucokinase is only present in liver
Why is it present? Okay, yeah,
cells.
yeah, that's true. All cells, and this is in liver. So why is it present in liver?
So because the glucokinase has lower affinity for glucose, only when there's a lot of glucose, it will be able to phosphorylate it.
Yeah. So glucokinase, it basically helps in trapping of glucose inside the liver cells because when we are having a diet, like when we just eat like having a meal, the abundance of glucose that we get in, most of the glucose we can't directly send it in the blood vessels. So they are getting mostly trapped inside the hepatocytes so that they can get converted into glycogen or like whatever like in that thing. So so which has the higher affinity, hexokinase or glucokinase?
So hexokinase is high affinity, less amount of glucose it can.
Yeah, comparatively they have this thing, low affinity, and like we which can has the like um higher km, not km, sorry, what is Vmax. So which do you think is for glucokinase and which do you think for is for hexokinase?
So for the hexokinase, since the affinity is high, Km will be low.
Mhm. Hexokinase is this, and this is glucokinase because the first thing is like Vmax is quite high here. Because as the concentration of the substrate increases, the Vmax, like the conversion capacity, also increases. But in hexokinase, only after like initially it increases, like in the lower concentration, it is easily able to trap the glucose. But as the glucose concentration increases, it fails to do so. So that's why it will reach the Vmax very quick compared to the glucokinase. So that's the difference between hexokinase and glucokinase. And one more thing, do you think glucokinase is always active? You told me that glucokinase is special, like it is present in liver, and it is present in the liver, and it takes a lot of glucose in. So, do you think it is always active? What if like I'm starving? Do you think it will be active?
No, so I don't think it is always active because uh, in low glucose, when the glucose is very low, we don't want to convert it. We don't want to break it down.
Hmm. So basically, in this hepatocytes, the glucokinase are normally present into the inside the nucleus. For example, this is the glucokinase. It is regulated. It is bound to a glucokinase regulatory protein (GRP). So when there is abundance of glucose taken into the cytoplasm, some of this glucose will move into the nucleus, and then this will activate this glucokinase, and they will take it out to the cytoplasm. So initially, this is a special enzyme. Like it is normally in its inactive form, it is present uh, inside the nucleus. But when there is a higher abundance of glucose, this some of the glucose goes into the cell and then into the nucleus, and they remove this glucokinase regulatory protein, and then they bring out the glucokinase into the cytoplasm, and there the process occurs. So this was the first part from this glucokinase, glucose 6 phosphate, and we have heard about hexokinase and glucokinase. Now tell me, as you tell, as you told that like when we don't have enough glucose in our body, and therefore like we can't store this glucose, or like we can't just let it be inside the cell. So we can't utilize this glucose. We have to leave this glucose as such. So how do you think these things will be inhibited, like these enzymes? Like obviously that should be like process in order to stop this process. If there is a like lower amount of glucose in our bloodstream, that means these cells should not take up the glucose easily. So there must be an inhibitor. Like who do you think inhibits hexokinase and glucokinase?
So if the ATP is quite high, maybe the cell is already having a lot of energy.
Yeah, but this step of glucose, that is, it's a irreversible step. Okay, like this is the first irreversible step. Like remember this, and then this hexokinase activity is stopped by this guy, glucose 6 phosphate. So when there is enough, like lot of glucose 6 phosphate in the body, this acts as a inhibitor, like product based inhibitor for this enzyme. So basically, this enzyme gets inhibited, and it won't convert more glucose into glucose 6 phosphate. So that's one thing. So do you think it will inhibit glucokinase as well? Okay, let's move forward first. I'll tell you. So from this glucose 6 phosphate, it is converted into what is the next product?
Fructose 6 phosphate.
Fructose 6 phosphate. It's the enzyme here is some isomerase will be there, right? What- So some isomerase is that, like it basically shifts some hydroxyl group here and there, and then from glucose, it is forming the fructose 6 phosphate. Now when this glucose 6 phosphate is converted into fructose 6 phosphate, then only this fructose 6 phosphate will act as a inhibitor of this enzyme. So always remember, hexokinase it is ubiquitous. It is present. It has a higher affinity of glucose, but the Vmax is reached really fast, and then it is inhibited by glucose 6-phosphate. But glucokinase, it is a special enzyme. It is normally present in an inactive state inside the hepatocytes in the liver. So when there is excess intake of glucose, this glucose goes and binds to the goes into the nucleus and takes out this glucokinase, and this glucokinase then converts this glucose into glucose 6-phosphate. And this glucokinase, since the affinity is like quite low and the Vmax is really high, this fructose 6-phosphate acts as a inhibitor of this. So when the body has a lot of fructose 6-phosphate, then only this glucose is getting inhibited. Glucokinase is getting inhibited. So always remember, glucose 6-phosphate is the inhibitor of hexokinase, while glucokinase inhibitor is fructose 6-phosphate. Okay. Now next step is from fructose 6-phosphate. Next product is
So fructose 1,6 bisphosphate.
1,6. Fructose 1,6 bisphosphate. In last class, I asked a similar doubt, like on a similar question. So what is the difference between bis and bi?
Oh, so bis means that the phosphate is attached to two different carbon or two different moieties. And if we say bi, to one carbon or one species, we are attaching the groups. Like in ATP, it is adenosine triphosphate.
No.
So to one carbon, we are attaching.
First thing is that bis and bi are like, I mean, your half the answer is right. Bis is basically that, yeah, two different carbons, two different atoms, two different atoms. But in bi is like, okay, I will draw something. So are they both bis? According to definition, like if the
Yes, according to what I said, they appear to be bis.
okay, so first thing is that this is bis, but this is bi. The definition here is that any group that is attached continuously, that comes under bi. So even if it's on the different carbons, it has to be in continuous. So if I keep one here and then keep other like in the next carbon itself, then it's a biphosphate. But if I keep one here and then next here, then it's bisphosphate. Bis 1,4, sorry 1,3. But this is bis, so bi 1,2 biphosphate. So the definition is basically in bi, it's just the continuous arrangement of an atom. So that's an answer. Like it's not the two different atoms, it's about the continuity. So we have fructose 1,6 bisphosphate. Okay. What is the enzyme involved here?
Sorry, sir, your voice was breaking. I couldn't hear.
So the difference between this bis and bi is that in bi, if an external group is present in continuous, for example, these are two molecules. This is bis.
No, no, sir. Actually, I understood the whole bis and why. Then you asked something, I was not
Okay.
able to hear that.
So what is the enzyme that converts fructose 6-phosphate to fructose 1,6 bisphosphate?
So phosphofructokinase 1, TFK1.
So like in the glycolysis, this is the most regulated and most important step in the entire glycolysis process. The conversion of fructose 6-phosphate to fructose 1,6 bisphosphate. So you told me about kinase. So you know the difference between kinase and phosphatase, right?
Yes, sir.
So what does the kinase do?
So kinase is a kind of transferase which is going to transfer the phosphate from, let's say, ATP, and it will add to the another substrate.
Okay, basically it
And
adds.
phosphatase,
Mhm.
yes,
Basically it
yes,
adds,
it adds phosphate,
and phosphatase basically removes.
removes phosphate.
Is there any exception to it? The question is like, do all kinase acts as phosphated? Like they like, do they always add phosphate?
So actually, in the lecture, one sir explained that like kinase is a kind of enzyme which is taking two substrate at a time. So one is ATP and one is the other substrate. Let's say here we have fructose 6 phosphate. So it is taking ATP from one and it is adding to another. So it is not always adding; it's also taking from someone. I
Okay.
can think of that.
So generally, yeah, like it's taking it, like the activity is similar, obvious, but um, there is this, like you have talked about phosphofructokinase 1, but there is this exception, that is phosphofructokinase 2. It has two domains. This PFK2. It has both the kinase domain and phosphatase domain. Sometimes it adds this phosphate and sometimes it removes it. It does, like, I understand the concept of like taking and then adding, but this is like it basically removes the phosphate entirely. So um, this fructo okay, let's talk about this step first. Fructose 6 phosphate to fructose 1,6 bisphosphate. This is the one of the most regulated and important, like this is the most regulated and important step of the glycolysis. So here the enzyme here is PFK1. So again, like first regulatory step was the first step, that when the glucose is converted into glucose 6 phosphate, that's the most, like this is the regulatory step, because it's only in one single direction, while conversion of glucose 6 phosphate to fructose 6 phosphate is, it's occurring in both the direction. So it's basically like not that important, the regulatory step. Then again, this is the second step, that is the most regulated one. So, as I told you in at the first level, that is glucokinase and hexokinase. Hexokinase is inhibited by glucose 6 phosphate, while glucokinase is inhibited by fructose 6 phosphate. The similar thing, when this like glycolysis like moves downwards, again this regulatory step comes in. So how does this cell know that, okay, like we already have like lot of energy, like we don't need that much, whether this process should go forward or back, like, or stop? Like how do you think this cell would know that they should continue this process or not?
So I think one is because of ATP,
Mhm.
because the end product of this glycolysis is production of ATP. So if there's a lot of ATP, we will not proceed. And other is maybe pyruvate and the substance from, I think citrate is there
Yeah.
from the TCA cycle.
So citrate and ATP, if they are like in like larger amount, so this thing will, like, are they the positive regulator or negative regulator?
So since they're stopping the enzyme, they are negative.
So they are basically the negative regulator. So once the cell has higher amount of ATP and citrate, they basically will stop the glycolysis. So what do you think is a positive regulator then? Like which thing will force this process to go forward?
So the thing is that I have the whole idea of what exactly happens, but I was not prepared for this lecture. So I'm not able to answer any questions.
Okay, like, okay. So you told me that ATP is the negative regulator. That means the energy in the cell is quite high. That's why the cell is stopping this glycolysis. But so what is the opposite? Like when this ATP
Okay,
is
okay,
broken
then
down,
one can be ADP.
AMP. It's not the ADP part. It's the AMP.
Okay, AMP.
AMP is the negative regulator, like positive regulator. And then one most important part, like apart, like the most strongest of all, the positive regulator is fructose 2,6 bisphosphate. This is the strongest, like it's the strongest regulator of this. When this goes and binds to this fructokinase 1, it basically fast forwards the reaction. Like it is like lot of this. I mean, this process will go at a higher rate. So this fructose 6 phosphate, it is basically converted to fructose 1,6 bisphosphate via the PFK1. This is the like most of the enzyme. Like for example, if you have 10,000 of it, 9,000 of the F6P will be converted into F16. But some of these molecules, like in thousand hundred about 100 molecule will be converted into fructose 2,6 bisphosphate, and the enzyme that works here is PFK2. So this enzyme will convert this F6P into F26, and this when it's the F26 is formed, it will go and bind to this PFK1, and then the rate of this reaction will be like, I mean, like it will be a lot faster. So now, so and as I told you, this is the most regulated and most important step of glycolysis. Most important and regulated, because as you can see, like lot of regulatory steps are here. Like positive is like AMP and F26, while negative is citrate and ATP. Now tell me, so as I was telling you that PFK1 PFK2, it has both phosphatase and kinase activity. So as I told you that this substrate is like extremely important for this glycolysis to proceed further, like faster and like more strongly. So what if I already have so much of substrate, like I mean like so what if I already have enough energy in the body, and I don't need this, like I mean okay, like this citrate and ATP are trying, like sorry, this AMP is trying to fasten up the reaction, but as I told you, this like F26 bisphosphate is the strongest regulator. So what if I don't need that regulator, like I don't need that much energy? So how do you think like I should stop?
So maybe we can inhibit the PFK2 so that it doesn't make phosphate
Like this
6
enzyme is already
phosphate.
present in our body, but it's mostly by stopping this conversion of this F6P into F26. So as I told you, it has two domains. One is kinase domain and one is phosphatase domain. So when in your body, when glucose is this is kinase domain, and this is phosphatase domain of PFK2, phosphofructase kinase 2. Now, for example, this is a cell, and as I told you, this is the insulin receptor. That's a RTK type. That is receptor tyrosine kinase, and one receptor is this is a glucagon receptor. Okay. Now the glucose level. So now the first condition here is glucose level is very high in the body. So what do you think will be released?
Insulin. So,
Okay, first let's start with this backward direction. Glucose is very low in our body. So this will release the glucagon, and glucagon will go and bind to its receptor. This is a GPCR type receptor. The insulin is RTK type, and this is a GPCR, that is G protein coupled receptor. It's a 7 transmembrane. As you can see, like it is spanning the membrane 7 times. So when the glucose amount in our blood is very low, that means it should not, the process should not go forward, right? So this thing, once this glucose goes and binds to this cell, it will give a like signal. It is a GS type. Have you like learned about this G proteins? GPCR, G stimulatory, CAMP cycle, PK.
Yes, yes,
Okay,
sir.
okay. So this GS cycle will get activated, and then CAMP will be high. When this CAMP is high, it goes and activates PKA. What is PKA? It's a protein kinase A. So what
So
did it?
protein kinase A.
So what it does is basically it will go and phosphorylate this domain, both the domains. Now once it gets phosphorylated, so what it does is once it gets phosphorylated, this what is this? Once this gets phosphorylated, this phosphatase domain will get activated. So now you know that, right? Like when this kinase, like most of the enzymes, like not in all the cases, most of the enzymes when they get phosphorylated they get active, but in some cases this phosphorylation leads to deactivation of that enzyme. Similarly, in this PFK2, that is phosphofructokinase 2, it has both the domains, kinase domain. Even though it's a kinase, it has both the domains, kinase as well as the phosphatase activity. So when we phosphorylate this PFK2 by PKA, this kinase domain gets inhibited, and this phosphatase gets active, and this will go and stop this conversion of fructose 6 phosphate to fructose 2 6 bisphosphate. So basically this process is stopped. And when this is stopped, because our cell does, like our cell in our blood, the glucose amount is very very low, and because of which the glucagon is produced, and this glucagon goes and binds to the receptor on the cells, and this thing will activate the kinase, and this kinase will go and phosphorylate the PFK2. Upon phosphorylation, this phosphatase activity of that kinase gets activated, and it will stop the conversion of F6 fructose 6 phosphate to fructose 2 6 phosphate, and therefore further glycolysis won't happen. Basically it will be slowed down, or like it won't happen. So this is it. So when we don't have glucose, kinase is formed, and the process is stopped. Now when the glucose is very very high in our body, insulin will be released. That means when insulin is released, basically it's telling the cell that move the glycolysis faster, like towards it. So when this insulin comes and binds here, it will like lot of downstream process will occur, and then our phosphatase will be activated. What this phosphatase will go and do is it will remove this phosphate from here and here, and when this phosphate is removed, this thing is getting it won't get activated, and then kinase gets activated, and this will move this process forward. So basically glucose high and glucose low, these are the two conditions. As I told you, this is the most regulated step, like fructose 6 phosphate to fructose 1 6 bisphosphate and fructose 2 6 bisphosphate. And here phosphofructokinase comes in 1, while this is fructokinase 2. So when glucose is high, insulin is released, then insulin receptor higher, then phosphatase is released, it acts on PFK2, and kinase domain gets activated. Sorry, kinase domain of it gets activated. Again, when the glucose is low, it will secrete glucagon. It will bind on this glucagon receptor. Then it will activate a PKA. It goes and interacts with PFK2, and the phosphatase domain gets activated. And then it will speed up glycolysis, slows glycolysis. So did you understand till here?
Yes, sir. Very nicely.
Yeah. Do you have any other doubt?
No, sir.
Okay. So now next thing we are going to do. Okay. Now we have to study till fructose 2. Sorry, this fructose 1,6 bisphosphate. Now what's the next product?
So aldolase breaks it down into three carbon compound,
Okay,
glyceraldehyde,
phosphorus.
3-phosphate, and DHAP.
So basically from so this is fructose 1,6 phosphate, and then from here two things comes in. Aldolase is here, and the reaction is both ways. So what is the compound here?
Glyceraldehyde, 3-phosphate, and DHAP.
So that is DHAP and glyceraldehyde 3 phosphate. So these are the two products. So the thing here is that we all know that in glycolysis, only thing that goes forward is glyceraldehyde 3 phosphate. So most of this DHAP is converted into glyceraldehyde 3 phosphate. So what's the enzyme here?
Isomerase, phosphate isomerase.
Phosphate isomerase. Okay. So from here, like both the process will be similar. Like this DHAP will be converted into 6,3P, and then the similar process is getting. So basically, it's twice, right? Now, till now, we know that from this glucose level to this which level? Fructose 1,6 bisphosphate or till glyceraldehyde 3 phosphate, we have not received any energy from the glycolysis, right? We have in turn we have given the energy. So like energy is utilized from this step. So here, like one of the ATP is utilized here, right? One ATP is utilized here, and then again from this phosphate, one phosphate is here, and then from F16 to uh, F6 to F16. So one is utilized here, right? So till now, based on the literature, we know that here two ATPs are getting utilized. So you can also say that this conversion of F6 to F26 is also utilizing one energy, but mostly they are not like, I mean, the as I told you, if there are like 10,000 molecules, like 1,000 molecules of F6P, 9,000 are getting converted into, like 9,900 are getting converted into fructose 1,6 bisphosphate, and only this 100 molecule are getting converted into fructose 2,6 bisphosphate. So it won't like people don't consider this phosphorylation much properly. Okay, now so we have like taken up two and okay, two ATPs are utilized right now. So till now there's just loss of two ATPs. Now from this step onward, we are like gonna extract like whatever energy that we have put in, like from this glyceraldehyde 3 phosphate, the energy is gonna be utilized. So from this glyceraldehyde 3 phosphate, which what is the next molecule?
So 1,3 bis phosphoglycerate.
1,3 bis phosphoglycerate. And what's the enzyme here?
Dehydrogenase.
So dehydrogenase comes here, right? Okay. So this is the first step of glycolysis where we get energy. So when this product, like when this substrate, that is glyceraldehyde 3 phosphate is converted into 1,3 bisphosphoglycerate, um, this like the energy here is converted like not into this ATP form. It will be like this NAD plus is getting converted into what is it is NADP or NADH.
NADH.
So it's getting converted into, like, most of the energy is utilized in conversion of NAD into NADH. And when, like not entire, like not the entire energy is converted, like some of the energy is used to do this phosphorylation. This inorganic phosphorus is also present. So some of this energy is used to add this phosphate to this glyceraldehyde. So there are basically three pockets here. This enzyme has a three pocket. One is for inorganic phosphate, and one is for NAD plus, and this is a substrate, glyceraldehyde 3 phosphate. Now, on the basis of this dehydrogenase, this enzyme, most of this energy is, like, when the substrate is, like, converted, like, this glyceraldehyde 3 phosphate is converted into 1,3 bis phosphoglycerate. And while the conversion is happening, this NAD is converted into NADH, which later goes into ETC to give 3 ATPs, right? One NADH is giving 3 ATP. So basically into 2 is 6 ATPs. This is for ATC chain. Okay. So, okay. This ATP can, so what's the process called when it goes to this mitochondria and does this electron transport chain?
So it is oxidation of this reduced form of electron carrier followed by phosphorylation of ADP.
So it's basically oxidative phosphorylation, right?
Oxidative phosphorylation,
Okay,
yes.
you have to remember this term. Okay, and then some of the energy that is left, it is like helping it. This inorganic phosphate is getting like added to this glyceraldehyde 3 phosphate to this phosphoglycerate. Now, okay, this is 1,3 BPG. Now, in certain cells, right? For example, RBC is one of the, like, okay. Does glycolysis occur in RBC?
Yes, yes, sir.
What about DTC?
We don't have any other organelles in RBC, so
So, for this thing to occur, like for oxidative phosphorylation to occur, what we need?
we require mitochondria and all the enzymes complexes.
And what is the second most important thing for ETC to occur? One is mitochondria, right? What is the other thing? Like, I mean, all the enzymes are present, like, but one thing is, like, really, really important. Any idea?
Yes, sir.
So, what do you think about oxygen? Is oxygen really important for this?
Yes, oxygen is the terminal acceptor of electrons.
Hmm. So, um, these two things are really, really important. So in RBCs, the, like, abundance of oxygen is present, but there is no mitochondria. That's why ETC won't happen, right?
Yes, sir.
One second. So where was he? Okay, yeah. In RBCs, this glyceraldehyde 3-phosphate is not directly converted into 1,3-bis phosphoglycerate. So there is one more enzyme here, mutase. It converts this glyceraldehyde 3-phosphate into 2,3-bis phosphoglycerate. Have you heard about this enzyme? Sorry, not bis, di. Have you ever heard about 2,3-DPG?
No, no, sir.
DPG or BPG, like even like it's same, I guess. Okay, so I'll show you this role. So like this is one of the most clinically important thing in RBCs. They have an enzyme called mutase. I forgot the entire name. There's this mutase that converts glyceraldehyde 3-phosphate into 2,3-bis phosphoglycerate, BPG or DPG. So what happens is, uh, so you know that hemoglobin, right? Hemoglobin has a porphyrin ring. So this is a porphyrin ring, and then if iron is present here, heme, so it's a hemoglobin thing. And hemoglobin is a tetramer, like do you know that?
Yes, yes, sir. It has beta and alpha. I think it's a tetramer quaternary protein.
So this is a heme, and the porphyrin becomes a heme, and then globin gets attached. And normally HbA type is found in our adults, like in humans, right? So this chain in normal conditions, when normally this blood basically carry like blood, the hemoglobin basically carries the oxygen and then releases into the in the tissues. So um, normally this what the 2,3-BPG does is it basically acts on this. Like what are the chains? Do you remember the chains? Alpha and beta, right? So this is the alpha chain and this is the beta. So what happens is, in the presence of 2,3-BPG, this beta chain cross links, and once the the oxygen is bound here, it basically crushes it. Like what happens is like this. So when they are interlinked, they are basically linked together, and basically it pushes the hemoglobin towards each other, and then the oxygen is released outside easily. So basically it increases the release of oxygen. And why do you think it is really important? Like normally, like when we are living here, so the use of this BPG is not that, like it's not optimal, like it's not required. But when we go in upper hills, like upper hill stations, like at a higher altitude, the concentration of this BPG, this conversion of this glyceraldehyde 3-phosphate is in BPG is quite high, to 3 BPG. And when this is high, as we all know that when we go up, hypoxia, the chances of hypoxia is quite high, because our body is not accustomed to like higher altitudes. But here the BPG here helps us to whatever oxygen that we are getting from outside, it helps in releasing this oxygen to our cells, our body, like entirely. So that's where BPG comes in. So this is like one of the most like important step in the RBCs. So conversion of glyceraldehyde 3-phosphate to 2 bis phosphoglyceride, and then to it will get directly converted into what is the next step from 1,3? Next is
Three phosphoglycerate.
3-phosphoglycerate. So this is the next step, 3-phosphoglycerate. So there is nothing important here. Okay, from here, one ATP is getting up. Here I think ATP is formed, right? Forgot actually.
Yes, yes, sir. ATP is found.
So ATP is formed here. So what is the difference between ATP formation here and ATP formation in the ETC?
So is it substrate level phosphorylation here, and that is oxidative phosphorylation?
Yeah. So it's a substrate level phosphorylation occurring in the So that's one of the most important difference between the normal glycolysis and the ETC. Here it's a substrate level phosphorylation. Okay, this is the phosphorylation. Okay, what after 3 phosphoglycerate is next product?
2-phosphoglycerate.
What's the enzyme involved here?
Enolase.
And what is the product here? Water, right? Hmm. And
Yes.
from this 2 phosphoglycerate, what's the next compound?
PEP. Phosphoenol pyruvate.
And then it's the pyruvate.
Pyruvate.
So what's the enzyme here?
So pyruvate kinase.
And here phosphoenol pyruvate to pyruvate.
No. PEP to pyruvate, I think it's pyruvate kinase.
Mm-hmm.
And then from 2-phosphoglycerate to phosphoenol pyruvate, there is enolase. And from 3-phosphoglycerate to 2-phosphoglycerate, we have mutase.
Okay. So here the water should be near the enolase part. Where is the second ATP found?
Uh, from PEP to pyruvate.
PP to pyruvate. Okay. We have already read this. Okay. Now let's. This is the final thing I want to teach. Is this. So okay. From PEP to. So you know this, right? Like how many irreversible steps we have learned till now?
3.
3, 2. So what is the first irreversible steps?
From glucose to glucose 6-phosphate, that's 1. And then from fructose 6-phosphate to fructose 1,6 bisphosphate, second. And PEP to pyruvate, third.
So these are the and what is the most important regulatory step of glycolysis?
The second irreversible, it's a rate limiting step.
All the 3 are rate limiting step, but this is the most regulated one.
Okay.
Now we have learned about the first 2, because in all of most of the exams they will be only focusing upon these steps. They will be either focusing upon um, exokinase and glucokinase, or phosphofructokinase 1 or 2, or this third enzyme PEP2 pyruvate. And this is the what was the enzyme here? Pyruvate kinase. Okay.
Pyruvate kinase.
So what was the inhibitor of hexokinase?
Glucose 6-phosphate.
And glucokinase?
Fructose 6-phosphate.
And inhibitor of PFK1? Like what will slow down?
Citrate and ATP.
Okay.
And also, if we have low amount of fructose 2,6 bisphosphate.
Okay, yeah. ATP and what is that? Citrate. And if glucose is high, the phosphatase activity of sorry, yeah, kinase activity of this. Sorry, phosphatase, not kinase. Okay. Now, the conversion of phosphoenol pyruvate to pyruvate is again the third most important step of glycolysis. So apart from this, they will target the questions of pyruvate kinase. So here, first thing is like substrate level phosphorylation is occurring. This is the one thing. Next is this pyruvate kinase. So again, this thing is regulated. So what if I don't need this energy? I don't need to proceed further. For example, it has passed the first step. It has passed the even the this second level regulation. But now at the end, it is not, it does not want to get um, does not want to proceed further. So who do you think will act as a substrate like of inhibitor?
So high amounts of ATP can.
ATP does not act here. So the inhibitor, the inhibitor here is fructose 6 phosphate. So basically fructose from here, is it? Like, okay. The fructose from that, from there, it will directly go and inhibit the fructose pyruvate kinase. Like it's stating that we are like, there is like lot of energy already, and so to stop this. So this is the third important mechanism. Like third important step of this thing. So it's a phosphokinase. Like sorry, pyruvate kinase is inhibited by fructose 6 phosphate. Okay. Now two molecules are molecular pyruvate is done, and it will go to the ETC cycle and everything. Like, Krebs cycle and all things. Okay. Now I have to mention what I had to tell you something. I had to tell you. So till now it is clear. Like am I like was I able to like give you like something new apart from what you know, which I
So
told you?
more than what my college professors have taught. It's in too much detail.
Yeah, like
Very nice.
so this how like in our most of our textbook it's all about the reactions and stuff, but these are the major things. Like mostly you don't have to focus on anything, you just have to focus on this three steps, and you have to remember the enzymes, you have to remember the inhibitors, and then you also have to remember this path. Like just like at the what basically happens, not at the pathway level, but at the physiological level. Why this thing is getting active, why it is not getting active. So the glucose, so everything, the entire glycolysis process is mediated by the glucose thing. So one more thing, okay, yeah, we have heard about, like we all know that like all these enzymes are present in the cytoplasm, right? Except this glucokinase, which is regulated by glucose when the higher amount is present. Okay. Now, this enzymes, hexokinase, glucokinase, uh, okay, let's talk this in a reverse way so that you can remember this. So this is a cell, higher amount of glucose is in your body. This glucose will need to be taken in. So mostly GLUT4 will act here. So always remember, like the specialty of like the five gluts. You always have to remember this five gluts because they are like really special, and yeah, like they are like mostly the important ones. Glut4 is insulin mediated. Like always have a like grasp on that. So when it's like lot of glucose is there, like it has to take lot of N. That's why glut4 will get activated. This is because of the activation of insulin. So when glucose is very high, insulin is secreted. It will go and bind to this RTCase. Now this RTCase, like lot of process occur. Like we have learned, like one process was activation of this phosphatase, wherein it will go and remove the phosphate from PFK2, and it will push the glycolysis forward. So this was one of the mechanism. Second mechanism was, so this enzymes that we see, hexokinase, glucokinase, and then this phosphofructose kinase 1, and then the last enzyme was pyruvate kinase. These four enzymes, like even though they are present already present in our body, but higher abundance of glucose in the cell tells us, tells the body that we need to produce this these four enzymes in like higher amount, so that the more amount of glucose can be taken in, more amount of glucose can be stored or processed. So this, even the like expression of these enzymes, these genes are present, like dependent on insulin. So once this insulin is activated, in few of the signals in the nucleus will express this enzyme very like readily, like in a high amount. So this was the final thing about glycolysis. Okay, I have told you about everything, right? Hmm, person- Okay. Now I'll ask some few questions with this glycolysis part. So like you were able to grasp everything, right? Like I mean, from the basic and physiological level as well.
Yes, sir.
Okay, let me ask few things. One second. Hello.
Yes, sir.
Sorry, I'm on call. One second. I'll get back. So I'll like ask you like few questions to you so that like you have I mean to check up on the topics that I've taught, and then if you have any doubt in this thing, you can ask. Like I have to leave in few minutes. I have an experiment planned right now. First thing I wanted to ask was, hmm, okay. Okay, one thing I forgot to tell was we have read about hexokinase and glucokinase, right? And I told you that from GIT track there are three monosaccharides: glucose, fructose, and what is the third one? Galactose.
Galactose.
So what is the preferred like substrate for this? Like I mean, what are the like out of the three like Which do you think? Will it be like will it be taking in?
So you explain that all of them will be going to liver, and from there, liver is going to decide how much to release in the
Hmm.
blood.
So in normally this kinase and exokinase both they don't have any partiality towards any of this substrate. They can take all of this substrate and they can easily phosphorylate it. So that's the thing. There's no specific. Even though if you think that it's a glucose, so it's like glucokinase, it is specific to glucose. No, and if you think that it's exo, and these are all the six carbon sugars, it's not like that. It is impartial to all of them. It can take any of it and then process it further. So that was one thing left out. Okay. Okay. Now tell me. Now let's talk about the physiological level thing. For example, you are in a fasting state. You are in a fasting state. Now tell me the pathway of how, like we all know that in a fasting state, the glycolysis won't proceed further. So what is the pathway that it follows? Like tell me from the receptor, like the hormonal level to the receptor level to the physiological change that will happen.
That you have just thought, sir.
Hmm.
Okay. So when we are in fasting stage, we know that the glucose is low
Hmm.
in the blood, and because of which the glucagon will be released.
Hmm.
And every cell is having the serpentine receptor, which is GPCR, specific for glucagon.
Hmm.
Upon binding, the downstream signaling process will occur, activating CAMP, secondary signaling molecule messenger,
Hmm.
which is which is basically has to inhibit the progression of glycolysis. So it will uh, inhibit the fructose PFK2
Why? I forgot about this.
because if so PFK2 has both the kinase and the phosphatase site, and if we do not stop it, it will produce fructose 2,6 bisphosphate, which act as a uh, very high positive regulator of the glycolysis.
So so this PFK, it will go and phosphorylate this, and then due to the phosphorylation, what gets activated? Kinase domain or the phosphatase domain?
The phosphatase gets activated.
Yeah, it gets activated. No problem. That is done. I have taught you that this enzymes are under the this the production of these enzymes are under the influence of insulin. So I have taught you that. There are three regulatory steps. These are the only three steps that are one way. Rest of the steps are like two ways. So once, like for example, if the body needs glucose back. So this glycolysis can occur in reverse direction as well, but only at these three steps the enzymes will be different. While the rest of the enzymes, which are they process in the both way, like they can even forward it and reverse it. But at these three steps, to take it in reverse direction, there are special enzymes. I forgot the names actually, but yeah, there are enzymes that will take it in a reverse way. So that's that. I've told you about the inhibitor pathways. Okay, exokinase affinity, you know that. PFK, ATP, and citrate, phosphatic. Yeah. Yeah. I've told you about 2,3 BPG because it will increase the oxygen release in the higher altitude. So this is the special enzyme that is only present in the RBCs. Okay, I've taught you that. Deaminase. Okay. So glycerylate 3-phosphate is the first molecule that has energy, and then from that only we take out the energy. Let's just like mitochondria and oxygen are required, but okay. Okay, this thing is okay. So normally GLUT4 are present in the vesicles, they are not on the surface. So once the insulin comes in, it will move. So that I have taught. And this. So do you have any doubt in this?
No, sir.
So like in the next class, like we'll be doing that. Like what's the topic going on?
Oxidative phosphorylation is completed, and now we are moving to photosynthesis.
So oxidative phosphorylation. So after glycolysis, it was oxidative phosphorylation, right?
No, no. After glycolysis, it was a Krebs cycle, and then oxidative.
Okay, so three things. So next class, I think we'll cover both ETC and then oxidative, and then photosynthesis has been started. Okay, okay, okay. Okay, in next class, I'll try to like complete those two things in a one go, and then make a few questions as well. Like actually, I need to go right now. Mm, so like I'll take this thing in the next class.
Oh, so sir, all the doubts that I have will also be taken in the
Yeah, yeah.
next
Like
class.
next class, like it will be on Monday, right? Yeah. Okay, yeah. In next class, it will be taken in the next class.
Okay, okay, sir. Yes.
Okay then. Thank you.
Thank you.
Bye.
Thank you, sir.